Perfect. Thank you, Michael. So I'm Elliot Holtham. I'm a co-founder of PROTEIC Bioscience. And on the call, we have Paolo, who introduce himself later in the presentation. I'm going to do the first part, and then he'll do the second part. Next slide, please. So just a little standard sort of forward-looking statement. My background, about 10 years ago, I finished my PhD. And ever since then, been doing startups, so serial entrepreneur in and around machine learning, artificial intelligence, and software. And really excited about this opportunity. If we go to the next slide. I think what we're tackling here is one of humanity's greatest challenges, and with every challenge is an opportunity. Today, there's 20 to 30,000 known diseases, and unfortunately, only about a third of the estimated diseases have a known treatment or an adequate treatment. And I think we've all experienced over the last 18 months or so firsthand how diseases, or particularly infectious diseases such as COVID, can disrupt all aspects of our lives. And I think this is one of the biggest opportunities right now, is the intersection of biology and machine learning. And it also happens to be about a $1.3 trillion commercial opportunity in the drug development market. There's a lot of players that have been tackling the space previously, and I think that the angle of machine learning and AI is really going to change the space significantly over the next 5 to 10 years. And a lot of tech companies, or some tech companies, are now also seeing that opportunity. I'm not sure if people are aware, there's been some high-profile news releases. So last year, Google started playing a little bit in terms of protein folding and understanding how proteins behave and interact. There was Nature, which is one of the biggest science publications out there, saying that it will change everything. There was a big BBC headline last year saying that one of the biology's biggest mysteries is largely being solved by AI. And not a huge press release, but about a week ago, Alphabet, which is the parent company of Google, launched a commercial venture, Isomorphic Labs. So, obviously, I think that the tech companies also understand that an AI-driven first solution has the potential to significantly disrupt the entire pharmaceutical industry, which is a massive commercial opportunity, as well as making a huge impact on our lives. Next slide, please. So in terms of PROTEIC's growth strategy, fairly straightforward. Our goal is to build the leading AI-based drug discovery platform. In the process, we're working on building a world-class multidisciplinary team. So because of the combination of machine learning and biology and pharma, we need people with backgrounds in different fields, and we need to build a strong multidisciplinary team. On the way, we're going to protect and expand our IP portfolio, collaborate with different science partners and commercial organizations for synergies. And really, at the end of the day, the goal here is to leverage the design platform to develop a portfolio of drugs which we own. Next slide, please. In terms of corporate leadership, our CEO is Kevin O'Neill, and we have a number of board members and also advisors that have significant experience both building technology companies from inception to exit or IPO. I think the track record that we have on our leadership team is really quite strong. And I think what we've done well here from the beginning is we've built the company, and we've built the advisory team, and we've built the board to look like a bigger company, so that we're ready, to have the structures, procedures, audit committees, all these things in place when we go public. Next slide, please. What's also really exciting for us is we have a founding shareholder group, which is out of the UK, which is the Frontier IP Group. They also co-founded, or the founding shareholder of another company I'll talk about briefly at the end, Exscientia, which just went public a few weeks ago on the NASDAQ for about $4 billion. So what we're really excited about having Frontier on board as a founding shareholder, and really using the learnings that they've learned over the last years with Exscientia to sort of help accelerate our progress and our growth. Next slide, please. In terms of our technical team, we're a mix of machine learning and AI experts, as well as sort of biology and bioinformatics people. We have a mix of sort of traditional, PhD level, postdoc level, and faculty members across the world as advisors. Our team is, sorry, just a question here. Our team is led by our Chief Science Officer, Dr. Eldad Haber, who's also a professor at the University of British Columbia here in Canada. Next slide, please. In terms of our technology, on the molecular prediction side of things, we're leveraging a lot of advances in and around machine learning and artificial intelligence, particularly graph neural networks. So if you think about, obviously, without getting into too much details, if you think about molecules or proteins, they're sort of connected structures together. So you could have atoms that are connected from a molecule, or proteins that are connected from amino acids, a basic building block. And you can think about these things being represented as a graph. And one of the latest advances in machine learning is basically how do we learn, or how do we make predictions on graph structures? And this type of breakthrough is really what's led to a lot of these new benchmark problems, such as the Google AlphaFold benchmark that was in the news. So we've been able to build our own technology in around these graph neural networks, which has been able to allow us to predict the properties of molecules on the computer, which helps us speed up the drug discovery process. Next slide, please. So like I say, we've built these specialized graph neural networks. There's a chart there basically showing some of our performances of our networks versus sort of state-of-the-art benchmarks. This particular publication is being accepted for the NeurIPS conference, which was one of the top machine learning, probably is the top machine learning conference in the world, in about 2 weeks from now. Our networks, we train them on GPU clusters. They can have tens or 100 millions of parameters, and we can use them to predict the physical properties or the interaction of different molecules with other molecules. Next slide, please. And now I'm going to switch it to my partner, Paulo, who's going to talk about our applications and our pipeline, and then I'll join at the end to talk about our current funding raise.
Thanks, Elliot. As he mentioned, my name is Paulo, and I work as the project manager at PROTEIC. I've been in the biotech field for about 8 years now, with the most recent which of experience being in actual protein biosynthesis and the business development around that here in Vancouver at a company called SignalChem. Elliot mentioned our growth strategy, and to look at that in a little bit more detail, we've developed a molecular design platform, so peptides and small molecules, with a two-pronged approach, one being targeted protein degradation and one being competitive or allosteric inhibition. I'll go into a little bit more detail about this later, but in either case, we want to use these to tackle some multi-billion-dollar industries shown here to develop our drug portfolio. Now, inhibition and degradation, they're the two tools that I mentioned that we want to take on, and to understand them, first, we start with inhibition, because this is actually where most modern therapeutic strategies are involved. And they simply work to turn off an overactive enzyme. A small molecule, in this case, shown in the schematic, that would simply bind and turn it off and thereby treat a disease. It remains a useful strategy today, which is why we chose to make it one of our tools as well. But the problem with another challenge here is that only 10 to 20% of disease-causing proteins have a targetable active site, the remainder of which a different solution needs to be come to. And that's where degradation comes in, where we simply get rid of the protein rather than inhibiting it. So looking in more detail at inhibition, we actually have a case study that we have already been working on. In this case, fighting pathogenic E. coli. Now, no matter your therapeutic strategy to try and treat a disease, it is very important to understand the mechanism of action to try and treat it. In this case, the bacteria, to try and simplify things, the bacteria secretes its own receptor, shown in this kind of in this schematic here. And once that receptor binds to our gut cells, it results in strong binding and adherence of the bacteria to our gut cells, resulting in disease progression. It follows then that inhibiting that interaction between the protein intimin and the protein tier would treat this disease. To do that, we used the native tier protein as a framework or a starting point for us to develop new artificial tier molecules that would competitively inhibit the binding to intimin, shown on the left here in blue, and treat the disease. A couple of different design approaches and strategies are shown here in pink and purple and red, with the highlight in yellow dotted lines at the interface between these two proteins. We can then take that information that we've gained from that to put the design through an iterative process. In this case, each new iteration represents an improvement in either binding interactions or stability. You can see in this case, we actually were able to remove a substantial portion of the protein tier here to make our artificial tier, which improves the stability and more importantly, actually the manufacturability of the protein with its smaller size. We can then put our design to a computational test where we see how it would behave in solution. One thing to take home here is that you can see that the interaction that we've created is so strong that the two molecules seem to actually act as one. Now, to understand targeted protein degradation, our second tool that I mentioned, we actually first need to understand degradation in general. This is a process that happens every day in every cell all the time to get rid of improperly folded proteins or old proteins, basically. And the way it works is our body ubiquitinates these misfolded proteins. Here, it would be this yellow protein here. And once it's tagged with a polyubiquitin, the proteasome or our cell's wood chipper simply degrades the protein. Once we understand this, we can actually use that in targeted protein degradation. Here, we can use an artificial or linker or adapter that would bring in proximity a target that would not normally be degraded towards these ubiquitinating enzymes. Then this process turns a previously undruggable target druggable because it no longer needs to be inhibited because it would simply be degraded. It's a very, very new technology. And one of the questions people might ask is, why would we choose this as a tool if it's that novel? A quick glance at what big pharma is doing shows just how important it is and how valuable they see it as. Each of Pfizer, Bayer, and Novartis just in the last couple of months, not even in the last year, just in the last couple of months, have put over $1 billion banking on target protein degradation. And the reason they've done that is because these degraders seem to improve on the stability of RNA-based therapeutics and also are able to tackle the undruggable targets that in decades of research into inhibitors has not yet been able to overcome. In either case, whether degradation or inhibition, the steps involved from a computational point of view are incredibly intensive. This is the challenge, but this is actually our strength. Elliot mentioned earlier briefly our work in artificial intelligence and in machine learning, and also the team that we've built around this who specialize in tackling this challenge itself. Now, I spoke a little bit about those two tools in our pipeline that I showed earlier. So I will also like to spend a couple of minutes talking about why we chose the particular fields of interest that we're going after. First, rare diseases. This is probably the fastest, if not one of the fastest growing areas of biopharmaceutical, in the biopharmaceutical industry, with a 20.5% compound annual growth rate. I will say it is unfortunate, the recurring revenue, because it's kind of the reason it's recurring is because these have genetic, a lot of them, majority of them have genetic causes, which mean it affects people for their entire lifetimes and therefore requires treatments for decades. and it's up and coming, with 58% of all new 2020 drug approvals designated for rare diseases, but only a small percentage of them having approved therapies. To example how it is, this business potential is not just seen by us. Just this year in July, AstraZeneca bought out Alexion, a small company specializing in rare diseases, for $39 billion. Oncology almost speaks for itself in that we all are pretty aware or may even know people who are affected by cancer. It has a large unmet need even today, even with all the research done on it, with 30,000 patients a year in the US alone, with a 5-year survival rate of 30%. That extensive research and studying into oncology is actually a good thing for us as well, because we can take all that structural knowledge and apply it to our platform and raise the foundation. For infectious diseases, I think COVID has put this on the map for, I mean, if it hasn't put it on your map, you probably been living in a hole somewhere, because the rest of the world for 2 years have been plagued with a particular infectious disease. But more than COVID, it has always been a constant evolving problem for the world to tackle, and pushing the potential market up to an estimated 86 billion by 2025, and affecting the entire world. And lastly, neurodegenerative diseases. The most famous of this would be Alzheimer's. I think everybody's familiar with. With an aging population, this is going to have more and more impact on everybody's lives and require more and more for the pharmaceutical industry to keep up with it. In all cases, you would have noticed at the bottom that a reason we're choosing these is also because we feel our platform is well fitted to tackle them. And with that, I think I'll pass it back to Elliot to talk about the trends in financing.
Perfect. Thanks, Paolo. So over the last year, it's been a very exciting and activity-rich year in the early stage drug discovery based on computational methods. I've just shown a couple companies here as highlights. We have Monte Rosa, which just IPO'd recently. They've only been around for about 3 years. A local company in Vancouver, AbCellera, IPO'd last year for about $4 billion. Other companies last year, C4 Therapeutics IPO'd in October. Arvinas, fairly recent IPO, in Chimera, also another IPO. And as I mentioned earlier, Exscientia, which has the same founding shareholders in the Frontier IP group in the UK, IPO'd recently in around just $2.5 billion. Next slide, please. So in terms of investment highlights, we're really excited about this. We're still an early stage AI drug discovery company, but we have an experienced and successful leadership team, as well as a deep science and technology team, which have successfully built multiple startups in around software, machine learning, and artificial intelligence. We're building this proprietary AI-driven technology platform with a strong roadmap with some massive opportunities in the $1.3 trillion drug discovery space, and are currently targeting a NASDAQ listing at the end of Q4 2022. And we feel it's an attractive sort of pre-IPO valuation point, especially relative to the peers, which I showed on the last slide, which are all trading in the single-digit billion-dollar range. Next slide, please. In terms of our funding, we completed a private placement earlier in the year, and we're completing our second private placement now, which I'll give details in the next slide, with the goal of targeting Q4 2022 for an IPO. Next slide, please. In terms of our current financing, which we're going to close December 15th, we're raising $2.5 million US at a price of 50 cents per share. Next slide, please. In terms of getting in touch with us, much of our leadership team, so Kevin and Brian and Eric, are all going to be at the event tonight in Florida. So, I'd like for you all to reach out and get to meet some of our team in person. Please check out our website at PROTEIC.ai. And please reach out to Brian Keane, who's in charge of the current fundraising and capital markets. That's Brian at PROTEIC.ai. Or sort of general inquiries, you can reach out at info@proteic.ai. And yeah, we're really excited about this opportunity, and please reach out if you have any questions, and please meet the people in person today at the social cocktail. Thanks a lot.
So we have some questions, Elliot. Do you want me to just present them to you guys, or do you want to go
Sure.
in?
Sure, why don't you do that? And
Absolutely.
or
So
we
this is
can
from
go through
Richard.
that.
What are the various assumptions that need to be made to untap an undruggable target utilizing your algorithm? We assume it is your unique IP algorithm. So 2 questions there.
Sure. So
what are
do
the
you wanna-
various assumptions that need to be made to untap an undruggable target utilizing your algorithm? And then I guess the second question is this your unique IP?
Sure. So I guess, I mean, Paolo can also elaborate on it, but I think the reference, the undruggable, was in the protein degradation side of things. So I guess the assumptions there are that we can find a linker that will bind to the E3 protein and also bind to the target protein, and that this behavior will allow the normal degradation process to still occur.
Mhm.
And there won't be any sort of disruption from that point of view. Anything else you want to add to that, Paolo?
Yeah. The undruggable term was used from the inhibition side of things, where people have tried to inhibit things for a long time and it hasn't worked. This new technology is involving in basically that linker that Elliot mentioned, connecting both sides. Speaking to more where our strength in IP comes in is because there are so many computational steps involved. Yes, there would be others that could try to do it, but it is actually very computationally intensive, which we feel like we can tackle.
All right, awesome. This question is for Elliot. Can you talk about some of the fellow science team that's not here, and your personal track record of previous AI applications and some of those successes?
Yeah, sure. So we've built, in the past, we've done a variety of different applications with a similar team. So our team is led by our Chief Science Officer, Eldad Haber. I was actually his postdoc about 10 years ago at UBC, and we've been doing startups ever since. Our last startup together was a startup in around, we did medical and defense applications called Extract AI, which was acquired in 2019. And between the two of us, we've been building out the rest of the technology team. So we generally hire sort of postdoc level people. And on the AI side of things, we have about 3 people in that space. So that a lot of them were either Eldad's previous students or people that we know. So we feel like we have a pretty strong machine learning and AI team. And we've also been building the biology and bioinformatics team. So we have a couple previous postdocs on the bioinformatics side of things. And then we also have some software developers that are helping us sort of build the platform out. So we believe strongly in the multidisciplinary approach. We're not just an AI company, and we're not just a biology company. We need to be at the intersection of the machine learning and biology. And I think that's where a lot of big breakthroughs happen when you build these multidisciplinary teams, because there's not one tool that solves these things. It needs to be solved as a group. And then we're also building out our advisory team. We have, I don't know if people are in the space, the University of Washington and the Baker Lab there is sort of one of the premier protein groups in the world. We have some of the people that came out of there that were ex-postdocs there. So we're really trying to build the best team that we can, and a world-class team, by going after the best people at the best institutions and groups around the world, and fusing that technology team with a great leadership team to grow this company.
All right. So Elliot, this next question is about, can you talk about your co-founder and Frontier IP and their previous successes and why they are partnering with you?
Yeah, so we were fortunate enough to be connected with the Frontier group early on in our journey. And after initial discussions with their CEO, Neil, we were really excited about their portfolio of companies that they're working with, in particular one company, which they were also the co-founder, which was the Exscientia Group, which I mentioned that just previously IPO. And I think, working with a group like this has, many advantages. Obviously, we can tap into their business development and their sort of the advisory network there. Also geographically, we're trying to position ourselves, around the entire globe with presences in Canada, the US, and the UK, and then also in Israel. So I think having the Frontier Group really helps us expand with some boots on the ground capabilities in the UK. And then, obviously having had a great success, with the Exscientia IPO earlier this year, working with a group that's already done it or being involved with something that's successful is always quite easy. Much easier the second time, I say, like, we're not obviously following exactly what Exscientia did, but just having people that were involved in a similar space with a great success story really helps us to sort of make sure that we're going in the right direction, the right opportunities, addressing the right markets, and helping us accelerate our growth trajectory.
All right, one last question here. What is the current valuation you're raising capital at, and how do you see your value increasing with milestones going forward? And if you can just go into what some of the key milestones are, that would be great as well.
Yeah, sure. So I think the current raise is 50 cents per share US, and there's currently around 52.5 million shares outstanding. For exact details and the documents, I'd ask you to reach out to Brian at proteic.ai. In terms of our milestones, we feel like we're at a strong inflection point. We feel that the current raise is quite attractive relative to some of the industry metrics and other comparables out there. And what we're trying to do is, sort of for the next milestones, is build up our pipeline. We've identified 4 sort of key areas. We've built sort of the first product that's allowed us, or the first version of the platform, which has allowed us to design and validate molecules. We think sort of the next milestone for us is getting more focused on a couple specific applications that we've identified and showing some, ultimately lab validation that we can bind or inhibit or degrade some of these molecules. And I think if you look at the comparables, most of these other companies are still sort of very early stage as well, and certainly pre-revenue. And we feel that, if we can get a couple strong candidates in our pipeline, that will really give us sort of that valuation bump.
Okay. Well, guys, thank you so much for joining us today.